Hi everyone! Now we are going to show you about M&M. Not this kind of M&M, but it is about mitosis and meiosis.
Mitosis, a process that duplicates a cell and all of its parts, resulted body cells or somatic cells, and they will differentiate into specific cell type depends on the arrangement in the cell. Meiosis, a process where a cell divides twice and creating four haploid cells, resulted sex cell, sperm and egg. Sperm and egg combines genetic material from two organisms. The differences between somatic cell and sex cell. The somatic cell involved in growth and cell replacement, while sex cells involved in reproduction. Somatic cell has 46 chromosomes, while sex cell has 23 chromosomes.
Function of cell division. There are two types of organism, that is prokaryotes and eukaryotes. For prokaryotes, there are bacteria and archaea. They divide their cell through prokaryotes fission. The function of prokaryotes fission is for asexual reproduction. For eukaryotes, protists and some fungi, yeast, they divide their cell through mitosis and cytokinesis. The function of mitosis and cytokinesis is for asexual reproduction. As for other fungi, plants and animals, they divide their cell through mitosis and cytokinesis too, but the function is a little bit different. It's for development, growth, and repair.
Growth and reproduction depend on controls over cell division. They are checkpoint proteins. When a checkpoint fails in a particular process, a cell will undergo mitosis uncontrollably and cancer form. Apoptosis. Apoptosis is defined as programmed cell death. Some point of time, the cell brings itself about destruction. Apoptosis and cell division. Cell division increases, apoptosis decreases. Both are normal parts of growth. An organism starts from a single cell that will divide repeatedly to produce many cells, and at the same time, some cells must die for the organism takes shape. When a tadpole becomes a frog, the tail will disappear as apoptosis occurs. The differences between benign cell and malignant cell. Benign cell grows slowly, while malignant cell grow and divide abnormally. The benign cell kept in home tissue, while malignant cell disrupting tissue surrounding physically and metabolically. The benign cell are not cancerous, while malignant cell are cancerous. Neoplasms. Neoplasms is an abnormal mass of tissue that results when cells divide more than they should or do not die when they should. It is also known as tumor. There are three stages in interphase: G1, S, and G2. In G1, the cells grow before the DNA replication and the chromosomes are unduplicated. In S, the DNA synthesis or replication occur and chromosomes duplicated. In G2, the stage form for completion of DNA replication. The cell synthesis the size of proteins that will assist in cell division, and the cell prepares to divide.
You guys too slow! Hey guys, what are you doing?
Oh, we're going for a job.
Oh, can I join you?
Yes, yummy.
Hey Aiman.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, class. How are you? Good. Okay, today we are going to learn about running.
Assalamualaikum.
Waalaikumsalam. What happened to you, Faiz?
Oh, I did yesterday and
May I see your wound? Show me your wound. Masya Allah, very bad. Luckily you still alive.
I'm very lucky.
Yeah, but don't worry, Faiz. This wound will heal later. Because
In prophase, chromosome condensed, tightly coiled and folded. They appear shorter, thicker and visible to naked eyes. Nucleus disappear. Nuclear membrane disintegrate. Central move to pole. Mitotic spindle form. Spindle fiber from each central attached to each sister chromatic at the kinetochore. Nucleic envelope then breaks up. In metaphase, two pairs of central align at opposite pole. Chromatic pair attach themselves. Chromosome lines up in a metaphase plate, which also known as imaginary plate. In anaphase, separation of centromere. Spindle attached to kinetochore begin to shorten. Spindle fiber continue to shorten, pulling sister chromatic to opposite pole. Sister chromatic are separated and they are known as daughter chromosome. In telophase, chromosomes have reached the two poles of the cell. Chromosomes start to uncoil. Nuclear envelope reform. Nucleolid reappear.
Hey Aiman.
Hey Paez,
Where are you looking for?
I'm waiting for my
Oh, how dare you don't tell me about your
Oh my god, is this a dream? She's so fine.
If you were a tiger, you'd be a cute one.
Honey? Honey? What are you talking about?
She's my girl.
No, she's mine. No, she's mine. She's mine.
Look, she's mine.
Choose me or him.
But I don't know him.
What? You don't know me?
What's going on?
Who are you?
I'm your Hana, sweetheart.
What? Who is she?
We are twins.
But actually, we are twins too. But how come you guys look the same?
because variation occurs in meiosis.
Prophase 1. The duplicate chromosomes condense and pair with its homologous chromosomes. Each chromosome contains genetic information. Then, the pair of chromosomes exchange information. The process is called synapsis. Metaphase 1. The homologous pair of chromosomes align at the metaphase plate. The spindle fiber then form. Anaphase 1. The microtubules of the spindle fiber shorten and pull the member of each homologous pair to the opposite poles. Telophase 1. Nuclear membrane reforms around the daughter nuclei. Each of them has one chromosome from each homologous pair. Then, interkinesis occurs. Prophase 2. Nuclear envelope breaks down and the spindle apparatus forms. Microtubules attach to the kinetochores of the duplicated chromosomes. Motor proteins drive the movement of chromosomes toward the spindle equator. Metaphase 2. All of duplicated chromosomes align at the spindle equator. Anaphase 2. Daughter chromosomes separated to opposite poles by the interaction between the motor proteins and microtubules. Telophase 2. The chromosomes arrive at opposite end of the cell. Then, a nuclear envelope forms around each set of chromosomes. The cytoplasm divides and new 4 haploid cells are resulted.
Do you know what is allus, random alignment, and fertilization?
Alleles. First, through sexual reproduction, offspring inherit new combination of alleles, which leads to variation in trait. DNA and alleles are highly similar, but they are different enough to produce alternative physical traits. Second, paternal and maternal chromosomes carry different alleles. Random alignment. First, the initial contacts between microtubules and chromosomes are random. Second, maternal and paternal member of a homologous pair can end up at either pole, the chromosomes from two parents. Fertilization. Combination of two gametes and fusing of nuclei are called fertilization. The fusion of two haploid nuclei produces diploid nucleus in the zygote. The differences between asexual reproduction and sexual reproduction. First, in asexual reproductions, genes are from a single parent, while sexual reproduction, genes are combined from two parents. Next, in asexual reproduction, offspring are genetically identical to the parent, while in sexual reproduction, offspring are genetically unique. Mitosis versus meiosis. Mitosis is a asexual reproduction and for the growth and repair of organ in an organism, while meiosis solely for sexual reproduction only. Mitosis occurs in somatic cell, while meiosis in gamete cell. Mitosis produces clones of the same cell, while meiosis produces variable offspring. Prophase in mitosis versus prophase 1 in meiosis. During prophase in mitosis, the homologous pair do not interact with each other, while prophase 1 in meiosis, the homologous pairs become zippered together and crossing over occurs. Anaphase in mitosis versus anaphase 1 in meiosis. During anaphase in mitosis, the sister chromatids of a chromosome are separated from each other, while anaphase 1 in meiosis, the homologous chromosomes are separated from each other.
Thank you for watching our slides.
And don't forget to like and subscribe.
Bye!